Hi, how are you?
Hi, Bradley. I'm the first one in.
Yeah.
Oh, good.
I don't know where everyone is.
Okay. Well, give me a minute. I'll finish breakfast.
Okay. Hi, Jesse, how are you? Good, yeah. Well, glad you could make it. I'm doing pretty good. Dick is finishing his breakfast, so we'll wait for him to finish up, and I don't know who else is going to show up. Oh, there's Vinay. Okay. Hello, Vinay.
Hello, sir. Hi.
Hi. See, Jesse said, "Okay, how are you?" So Vinay, did you pick a paper, or are you going to wait for next week on that?
No, sir. I have my presentation ready. It's a short presentation. It's a small idea, so
Okay.
it's about. It'll take about 10 or 15 minutes. That's it at max.
Okay, that sounds good. We'll wait here for a minute, and then we'll start. Anything going on, Jesse or Vinay, that you'd like to mention, or?
I'm back.
Okay. Jesse, is there anything that happened this week you'd like to mention to the group? Okay, yeah. Yeah, thanks for going over the blog post. Yeah, I saw the edits. Thank you. So the blog post is on its way out. I'll post a link. I'll send it out. I'll send it out to the group when it's posted and finalized. Jesse contributed some edits. I saw Dick was contributing, and Vinay was contributing, so that's good. And it looks pretty good. It's very long for a blog post, but these are the types of things that we can maybe build upon in the future, and maybe some people will find it helpful. So we had, you'll see the blog post when it's finished. It'll have some links, and it's going to look pretty good, I think. I could have used the diagram, but it's always hard to make the perfect diagram. So I'd like to do one more post this year, or this, by the end of the year. Depending on the topic, we'll have to think about a topic that we can do another one on. But we'll look and see how this blog post. Yeah. So Jesse said he was trying to find a visual. Yeah, it was a little hard for me to figure out the perfect visual. So, I actually taught the Sci-Fund course on science outreach, and we had a whole section on blogging. And, one of the things they say is, use a lot of visuals. But sometimes it's hard to find the right visual. So, but so I think it was a good experience. And we'll see how this one does. And then if anyone has any ideas for a second blog post, we can talk about it later. So today we're going to do two things. I have a talk on GANs, which are Generative Adversarial Networks. It's not something that probably, maybe Vinay's heard of it, maybe Jesse, but it's an up-and-coming technique. I'll talk about. It's pretty interesting. And then Vinay has an idea he wants to pitch to the group.
Yeah, even my research paper is roughly around GANs itself, like mostly biospecific GANs, how they used adversarial techniques to synthesize retinal images. That was the topic of the paper. So, yeah.
Yeah, that should fit right into the talk, like at the end of the talk. So we'll just continue on to that. And then Jesse had a question about where the blog post would be. Yeah, I'm going to put it on the node, and then I'm also going to cross-post it to my own blog. And so, we'll have two places where it is, basically copying it over to the other blog and having twice the audience, I guess. Probably some overlap. But anyways, so I'm going to start with this talk on GANs. Let me share my screen. And again, this is just, I'm not super familiar with this topic, but I wanted to bring it up to the group because I think it's pretty interesting. It's a pretty new topic. So can everyone see my screen, my slides?
First, we don't have the interesting.
Okay.
Yes, sir.
All right. Let's see. Presentation. All right. So GANs are generative adversarial networks, and I put developmental above because I'm going to talk about some examples from development at the end. And there aren't that many examples, but this is an up-and-coming area, as you'll see. So generative adversarial networks is a pretty big mouthful of words. Let's see if we can break that down. So our GANs, or generative adversarial networks, consist of multiple deep learning networks. So we talked about the deep learning networks here at the bottom, and these are, of course, differentiated from neural networks because they have multiple hidden layers instead of just one. And so we talked about maybe a little bit about the benefits and drawbacks of that in earlier talks. Basically, it allows you to have higher resolution to increase your feature space, but there's some drawbacks in terms of the type of input data that you might use that are appropriate for the deep learning networks. And you have an increased chance of multiplying your error. If you have a data set with some systematic error in it, you can really screw up your task by just throwing those data at the deep learning network. So there are ways, there's some drawbacks to using deep learning. I guess the advice that I would say a lot of people seem to give is just don't throw deep learning at a problem, just as you wouldn't throw a t-test or a regression at a problem. So anyways, these GANs take advantage of these type of deep learning networks. And as you'll see, they use them in a very innovative way. And they have something called a generator and something called a discriminator. and I'll talk about what those are in a minute. So a generative model, and I know a lot of you are familiar with them, but maybe the jargon is a little fuzzy. What they do is they generate instances of a type or a class using some underlying distribution. And I show a slide of this is actually of evolution of peppered moths, which is a common example in evolution textbooks, which has not exactly been told the full story, but that's an aside. But the point is that you get this generation of variants at different generations, and it changes the the composition. What's generated changes the underlying distribution. So in this case, you have a lot of white moths and one darker moth. And in this case, this is, of course, when selection is applied. But a generative model is, in this case, it's a deep learning model. You're generating a lot of brown moths, but only a few white moths. And that's because you've changed the underlying distribution of the what's being generated. So you have a bunch of individual instances, and they're based on sort of a template, and then but the actual instances are variable based on that underlying distribution. So a neural network is technically a generative network, but also a genetic algorithm is also a generative model, and a cellular automata is a generative model. So that's the type of thing we're dealing with. And then a discriminant model or a discriminative model is where you generate a classification using an underlying distribution. So in this case, instead of generating moths or different colored moths, we're actually trying to classify butterflies. And so we can classify them here into different genera or, other sorts of classifications, and, based on their physical characteristics. So their wing color and their wing shape. And so discriminative models, regression would be considered an example, but also some sort of taxonomy. That's what we're talking about when we talk about a discriminative model. And it's very different from a generative model. So if you're interested in learning more about these different parts of a GAN, there's a link here. This is a Google Developers tutorial, and I can share that link later on. It's basically Google Developers. They have a bunch of tutorials on different topics, and they talk about the structure of a GAN. So I'm going to talk about it in a couple slides, but that's another resource. So GANs are actually pretty new. We have to go all the way back to 2014 to find the first instance of a GAN in the literature. Ian Goodfellow, who works at Google Brain, and he doesn't look too old here, and he hasn't, aged that much since, but he submitted a paper to NeurIPS. It was a paper in their proceedings called Generative Adversarial Networks, and that lays out the whole model of a GAN, and the different parts of it and everything. And then since then, there's kind of been an explosion of people applying this type of model to different types of data. It could range from even like doing things like generating artwork to biology data, next-gen sequencing data, and there are many different potential applications. But one more recent paper that's come out, and this is from the OLCR proceedings. OLCR is another machine learning journal or conference. They call it Many Paths to Equilibrium. GANs do not need to decrease a divergence at every step. And so what's interesting is that GANs were always formulated a little bit differently than most machine learning models, in that instead of talking about like the algorithm converging upon a solution, they talk about two different sort of ways of viewing how a GANs can be considered to be sort of optimized or successful. One of them is that the generator and the discriminator play this game, and they model it as a game theory or a game theoretic model, and they look for the Nash equilibrium. So Nash equilibrium is actually from game theory, and it's the idea that neither player can reduce costs without changing the other player's parameters. So in other words, you have a generator and you have a discriminator, and they're both competing to sort of outwit the other. And you'll see how that works in a minute. And so they kind of hit this Nash equilibrium where neither of them can sort of defeat the other one, and that's how you have an optimal model. But that view, of course, is contrasted with something called divergence minimization, which is where, and you'll see how this is applied, where the generated data have the least amount of difference from the classified data. And so they have this measure of, like, you generate your images, and then you classify your images, and there's this minimal divergence between the two, and that's how you gain some understanding of your GAN being efficient or successful. And so both of those are competing views, and I think it's interesting, though, and this might be something for Jesse to think about, that there's a lot of game theoretic sort of implications for this type of model. So, we probably didn't think when you logged on this morning that we'd be talking about game theory, but we are, and it's not very clear what's going on in this type of model, because we've only had GANs for about 5 years, and so people really probably don't even understand the models that well at this point. But it's an interesting path.
Bradley?
Yeah.
There's some similarity between GANs and simulated annealing. Are you familiar with that?
Oh, yeah.
Method?
Yeah.
Yeah, because simulated annealing is not always finding the best solution. It bounces away from it sometimes on the assumption that there's another path to a better solution than the one that's currently on.
Yeah. Yeah, so for Jesse and Vinay, are you familiar with annealing, which is a physical, it's like where they take a piece of metal and they put it on some irons and they heat it up, and then they get it really hot and they start banging it with a hammer and bending it and shaping it. That's what they call annealing.
And you cool it.
Yeah. So the idea is they're simulating that process. What was that?
I'm familiar with that, but not any kind of simulated version of that.
Yeah, the simulated annealing goes back to the Montrose work in the 1950s. Okay? It basically started when computers started. So it's been around for a long time, and it basically, you have a temperature, and you keep heating up the system and cooling it alternatively, where this temperature parameter locks into lower energy states. Then what you do is you slowly decrease the temperature over time, so you should lock into a solution. So it's usually used for optimization problems. Yeah. So it's a very obscure. At least now it's an obscure technique. But I learned about it in school, so I don't know if they cover it that much. Now with machine learning, it's like everything is new again. Like people are rediscovering this stuff, and so GANs. Let me go over. Well, first of all, let me go over how they've applied this to art. I think this is really fascinating, and then I'll talk about the actual structure of the algorithm. So this is what they call, this is one example. It's called GAN-Go, and it's a play on Van Gogh. And what they're trying to do is they're trying to create art with GANs. And so Van Gogh, of course, is an artist with a style. And the idea is you take that art and you take images that are Van Gogh paintings, and there's an assumption that there's an underlying style. Art critics will tell you that, but they won't quantify it or anything. And then you have this generator, which generates either some sort of random array or some sort of fake vector, fake data, like made-up data that you plug in. Like it could be a random number generator, or it could be like some, systematic noise or something like that. And then you combine those two and you feed them into this discriminator. And like, this is just kind of a shorthand for this. But what you're doing is you're feeding that in, and then you're trying to extract sort of the artistic style, and then use that artistic style to like generate new versions of Van Gogh paintings, but with the same style as Van Gogh. So it's like, the pictures up here, they look kind of familiar, but they're not. They're sort of eerily similar to something you've seen before, but they're not exactly the same. You can see because it looks a little, trippy, the lines and everything. That's because the computer generated these images, and it based it on a style that learned, and then it's generating new instances. So it takes a Van Gogh, it takes the style, and then it says, okay, make me a Van Gogh, and it'll make a Van Gogh-like image that has, the same stylistic properties, but it's a little bit off. And they call that sort of thing style transfer. And it's actually very fascinating because it's been actually quite successful. A lot of people do this. And there's another example, a neural algorithm of artistic style. It's an archives paper from 2015. So they're, using this very successfully in the art space. People fail a lot, and there's a community of people who, try different techniques, but I think in terms of creativity and like sort of applying GANs to a interesting problem, I think this is definitely a place that people should check out. I had a comment in the chat here. Okay, so Dick sent out a simulated annealing citation, so check that out. So yeah, there's a whole literature now on these artistic works that were created by GANs or using GANs, and so I don't know. I mean, maybe it's worth maybe just interesting art, but it might just be like something that people can learn from for other domains. So I'm going to go through like a GAN what the components are now. So I think it's probably worth doing because it's pretty confusing when you first encounter it. So up in the top left-hand corner, you have what I referred to as the generative model earlier. And so this could be what you do is you feed in some sort of pseudo data, some sort of fake data. Again, it could be a random number generator sending in information. It could be like systematic noise in 2 dimensions, like static on a television array, a television monitor. It could be a bunch of different things depending on your problem. And then you have training data. So you have real world data. It could be any type of data you can think of. So for the example from GAN-GO, you would have in the training data category, you would have actual pictures of Van Gogh paintings. And you train the algorithm on what those look like. The generative model, meanwhile, might be some sort of random noise or some sort of scrambled image. And so it's learning, basically, it's trying to fit the training data to some sort of what they call latent space. And then it's feeding it into the discriminative model. So the discriminative model picks the instances from that latent space and distinguishes things that are real from things that are fake. And so this is a procedure that's used to sort of build a sort of you could call it a landscape. I did a deep dive on Saturday into latent spaces, and I found out that they're related to multidimensional scaling. So latent spaces are actually low-dimensional manifolds or spaces that are derived from very high-dimensional information, like a bunch of paintings. And you're creating like a computational representation that's, has essentially all different combinations. And then the discriminative model is picking out the genuine article from a bunch of fake articles. And the adversarial part of the name GANs comes from the idea that you're taking real data and you're also taking data that are maybe not real or some other thing, and you're making the discriminative model choose between those two options. So it's fake or it's something else, or it's real or it's something else, and it's supposed to improve its training over time. So it generates a classification. It could generate a binary classification, or it could generate something more complicated. But you also have backpropagation. So these discriminative models don't operate on the basis of magic. They operate on this backpropagation. So the performance of the discriminative model is evaluated over time, and it's improved based on how it's performing on its classification task. So it might start out basically spitting out a bunch of nonsense as real images, but then over time it might pick up the real instances and successfully not only give you real instances, but things that are close to the real instance. So this is just some math here. So this is an example of an unsupervised GAN, and this is from a reference that we'll talk about later. But they use a minimax function where they look, so they're taking X as an image and Z as a latent vector, and they're performing an operation on that. They're basically taking the sum of that, and they're using their minimax function to find the best fit to that. So here are two examples from faces. This is an example where they take it, this is an input face, and they're recovering a latent space, and then they're recovering an image from that latent space. And so here you have almost a perfect match in this example. But in this example, you encode this face to a latent space of like faces. I don't know if you can put a bunch of different faces in and get this particular face, but the idea is you're taking the face, encoding it to this Z prime latent space, and then recovering an image from that latent space. In this case, it's a little bit fuzzy, it's not recovering exactly the same face, but it's a little bit distorted. That's because you're putting in this noise, this fake data to fool or to attempt to fool the model, and that's what you usually get. So you can usually get pretty good performance, but not always. And then here's some citations, and I'll make the slides available later. Some citations that might be useful. I think we have another question in the chat. Oh no, we have Dick coming back in. Okay. All right, Dick, are you back?
Yeah, I need a sledgehammer for my router.
Okay. All right. Let me go back to the presentation then. So I'll make these slides available later, but this is basically the idea of a GAN. And so, again, you have this latent space, and I've made it like an oval or an ovoid shape to sort of represent maybe a little like a two-dimensional latent space instead of a one-dimensional one. But the basic idea is you have this latent space, and it's kind of like a fitness landscape, or maybe like some sort of problem space that you would encounter in a regular machine learning algorithm. So you have this latent space of possibilities, and then you have this concept X, and you have the reproduction of the concept. And this is the input of the model. This is your desired target. And X star is the reproduction, or the one that you get out. So in this case, it would be this face here, or this face here, which would be X star. And then you evaluate one way to evaluate your algorithm is to use a reconstruction loss method, which is just basically simply the difference in distance across the latent space for these 2 points. So if you can get really close to your original concept, and that's great. But oftentimes the reconstruction loss is a bit more. And the thing is, you want to minimize this reconstruction loss as you run the algorithm. And that determines your performance and they don't call it convergence, but basically would be the convergence of it. And so you synthesize fake examples, you give it some data, you get a random noise vector, which is a latent space. You have some raw input, which are the images, and then that's your starting point. And then you run the algorithm So you can minimize the reconstruction loss, which is x minus x star. That's basically it. Then they have strategies that you'll run into in that literature. So one of them is to use something called an autoencoder. And so an autoencoder is a way to take the input and create the code, which is in the middle here, is this latent space creation. You're encoding your data, and then you're decoding your data as the output. And so, I mean, with GANs, you can go very deep into this area and, come up with some pretty innovative solutions. But I just wanted people to know that autoencoders exist and that it's a strategy. But now we're going to talk about examples from developmental data. And so this may make this a little bit clearer in terms of how it's used and how it's useful. So this is an example from a recent paper. These are cells that have been generated from a GANs model. So these are based on real examples of embryos, that's human embryos that have been fed into this algorithm, images of human embryos, and they have them at different stages of development, 1, 2, 4, and 8 cell, which is pretty early in embryogenesis, but you have these pretty simple examples for the algorithm to sort of classify. And then you have these two performance curves. One is the generated data, and then the other one is the actual data. So we'll talk about that here in a minute. But I wanted to talk about maybe two papers before I talk about that one. So the first paper is an application of GANs to gene expression and single cell classification. So in this case, they took a bunch of cells that were differentiated cells that differentiate in development. So they took motor neurons, hemopoietic cells, and then looked at hemopoietic cell differentiation in different ways. And so they took a bunch of different cell types, and they required scRNA-seq data, which is a form of sequencing RNA, so that you can map it back to the genome. RNA-seq is a very complicated technique, but it basically takes transcripts in the cells with what kind of genes they're expressing, and maps them to a whole genome map. So, you get a fragment of RNA out of the cell, a transcript, and you map it to the genome, and then you find out your abundance of those different transcripts, and it tells you about what the cell is expressing, how much of it it's expressing, and so forth. But it's sequence specific, and it happens all across the genome. So it's a very good technique for understanding differentiation or other types of perturbations that you want to introduce. In this case, they applied a GAN to that type of data for a bunch of different cell types. So they used epidermal cells, neural cells, hemopoietic cells, and then they used a generative model that simulated realistic types of RNA-seq data. So they basically simulated some data and put it in as in the generator. So the model you gives the model some input data. It's just fake RNA-seq data. It looks like RNA-seq data, so it has the same structure as RNA-seq data, but it's just made up. And you put it into the generative model, and it's generating different possibilities from that alphabet. And then you're matching it up with real RNA-seq data. And then you take the two and you generate these models, and then they do some mathematical projection of the data. But in this image, you can see that there's a real data set and a generated data set. So the real data set's in blue, and the generated data set's in red. And you see for each example here that they give, the real data and the generated data, for the most part, they overlap. You get some examples here where you get some outliers of the generated data, but you also get some outliers of the real data for motor neuron differentiation. But by and large, they match up. And so that's a good sign that you can use this for more than generating art. It's also a good sign that it's probably useful in terms of
looking at maybe like your data as opposed to like the null hypothesis. So I mean, there are different ways you can use these models, and that's one of them. Another one, of course, is this generative models from tissue organization with supervised GANs. So in this case, they're using a variant of the GANs model where they're using supervised data. They're using labels. In this case, they're building generative models of electron microscopy images. But in this case, they didn't just give the image data. They annotated the position of cell membranes and mitochondria in the images. So they were able to actually mark them off and give that information to the algorithm. And then they were able to use a specific procedure within the GANs framework, and they produced realistic images using a supervised GAN. They synthesized the label image, so that was your sort of your training set, and then they give a noise image as input. So they have a noisy image and a labeled image as the the noisy image would be in the generator, and then the labeled image would be as the input data. And then you bring those together in the classifier and see if the classifier can distinguish between them. The label in general provided supervision for classifying the EM image and even EM image synthesis, which is the synthesis of the image or the recovery from the weight in space. And then the full model generates label image pairs. So you're able to generate an image and you're able to generate labels to put on top of those. And remember, the images that it's generating aren't real images. They're not spitting back out real images. They're generating new possible images, but they're keeping the labels for both the membrane and the mitochondria. Another example is the one that I showed in the sort of the intro slide to this section. It's called HemiGen, Human Embryo Image Generator. And this case is, so this is, again, these 1-cell, 2-cell, and 4-cell examples where they're taking real microscopy data, they're generating some noisy input, and you're creating this latent space, and then you're recovering images from the latent space that are that look real. And the way we sort of evaluate that is we use input data and then we look at that versus the generated images. So in this case, they have histograms, and they didn't put labels on these histograms, but I'm assuming that it's like some sort of count. And they have these histograms for like the 1, 2, and 4 cell. Basically, the thing you need to pay attention to here is that the blue function is the input data, and then the red is the generated image, and you can tell because it's smoother, but that for the most part overlap. There's some parts where they don't overlap, but they basically overlap where it counts. And that's, I guess, a sign that the algorithm is doing well, that it's recovering very, similar images to what's being put in as a input data signal. And so in this case, they use a deep neural network. They trained a deep neural network so each part of this GAN is a deep neural network, and they're able to generate these 1, 2, and 4 cell images. And they claim a misclassification rate of 12.3% for the generated images. So this overlap here represents an error rate. 12.3%, depending, considering what we're trying to do here, may be pretty good. But there's no literature that really gives a good error rate. Good error rates are usually like within 1%. So this is kind of high compared to other types of machine learning or other types of pattern recognition. But considering what we're trying to do here, it might actually be pretty good. And then finally, there's this paper on contour extraction from Drosophila embryos based on GANs. And so this case, this is like some conference paper. In this case, they're generating images of in situ hybridization. So this is where they take an antibody and they stain like some tissue, and they look at like where the stain, settles into the
tissue. So in situ images kind of look like fluorescent images, and they have a fluorescence in the stain, but they show these areas of interest. So say you want to stain for some, some neurochemical in a tissue or in a cell culture. You would do this in situ hybridization process, and it would basically bind to the places where this compound is found, and then you'd put it under a microscope and you'd see these large regions of fluorescent activity, and that's where you would, that's what you'd be interested in. So they used images like that. So you have like basically Drosophila embryos with these in situ stains in the embryo, on the embryo. So it's not the whole embryo, it's just where this thing of interest is located. It could be a gene being expressed, it could be protein or whatever. And then they're using these images, they're doing a very high-tech way of analyzing these images. And they used a conditional GAN, which is, again, another variant of the GAN model, where they use a Bayesian filter to generate contour maps. So they're taking the in situ images, which are kind of have fluorescent information in them, have anatomical information, and then they're generating contour maps, which I assume are like concentration gradients of the different, of the hybridized fluorescence. They didn't have any images in this paper, so I don't really know for sure what that looks like. But and then they based this on a Drosophila embryo benchmark data set. So they used a benchmark data set as the input data. They used some fake images as the generator, and then they put that into a latent space, and then they selected images that looked to, contour maps that looked to match the input data. And so, again, in this case, you're trying to extract contour features from images, based on these adversarial examples and real examples, and it's supposed to sharpen the result. And so that's all I have for that. I mean, it's a pretty, so I guess what I would say is it's a pretty new area. I picked those examples not because they were the best, but because those were the only ones I could find. And so there's definitely a lot of room for improvement in this area. If it's something I think that if someone just wrote a paper on and mastered the GAN technique and had the right data set, they could really do something impressive with it. So are there any questions or?
Yeah, Bradley, getting back to your title, multiple, or were you talking about, when you say multiple, you mean multiple layers, is that correct?
Which slide was that?
Way back, first or second slide.
You mean maybe the second slide? Oh, let
Yeah,
me share my
probably.
screen, yeah. Probably work better. All right. You mean this slide here?
Yeah, I think so. You use the word multiple. No, by multiple, you mean multiple layers. Is that correct?
Yeah, multiple hidden layers.
Yeah, you see.
Well, actually, a multiple. Yeah, so with this, okay, when I say multiple up in the top here.
Yeah, that's what threw me a bit.
This is the deep learning network.
Yeah.
What you're using in this model here. This is a GAN in its full glory. You're using a deep learning model here, and you're using a deep learning model here.
Ah, okay. So you're using
yeah. So in the sort of the typical GANs, you would have a deep learning model doing this generation of data, fake data, right? So you're giving these adversarial examples.
Oh, so it is a small network of deep learning systems.
Yes. Yes.
Okay, so that's where my confusion was. So you actually do need multiple deep learning networks.
Yeah.
It's sort of like a meeting of minds, huh?
Yeah. It's like they're deep learning networks are two different functions. You're training one to discriminate and then one to generate a bunch of
Adversarial
basically,
examples, yeah.
in a way, then they're basically interacting deep learning networks.
Yeah, they do interact.
Okay.
Not directly, but they're basically.
Now, you've got a very simple graph here, or network of deep learning networks.
Yeah, it's simplified. Yeah.
No, it's simple. But what about if you consider a network and each node is a deep learning network?
Yeah.
Has anyone looked at that sort of thing?
Oh,
In other
right.
words, an array of basically an array or a network of connected deep learning networks.
Oh, yeah. Well, they're pretty hard to implement. Like, the computational power you need for one is pretty high. So, like, having a network of them like that is probably, yeah, I don't know if they have the computing power to.
Well, you do the old trick of networking computers.
Oh, that's true. Yeah, I guess you could do that. I don't think anyone's looked at that, though, no. And it's
okay. What I'm saying is, in a way, to stand back from it here, you're saying that you can have models of you can have neural networks,
yeah.
each of which is part of a network of networks.
Yeah.
And go way beyond this simple model here, and you might get some cooperative behavior between them that's quite different from the behavior of an individual.
Yeah, you could have that. Yeah, I mean, this is like, I guess, just something that they came up with, like, to solve a problem originally. But there's no reason why you couldn't do something like that. Or I was thinking when I put this together that you could use not just deep learning networks, but you could use like other types of models. Like this could be a genetic algorithm, and this could be some sort of like Bayesian filter. And you could use it, I don't know, maybe a lot better, or might not be good at all. But
yeah, but I'm thinking of each one as a sort of computer, and then you've got a network of computers that are sending information back and forth between each other. Yeah. And each one may not be, each one is doing its own thing. It might be different in various ways.
Right.
Okay.
Yeah. Yeah, that would be an interesting experiment. I don't know if anyone's done that, but
so a sociology of neural networks.
Yeah.
It's about time they talk to each other.
Yeah. So, Jesse or Vinay, I know Vinay wants to present. Maybe we'll, let's see what we have in the chat. Okay, so Jesse said, thank you. I took some things away from that. I heard of GANs for a while, but I didn't use them. Yeah, they're pretty new, so it's a pretty new area. So why don't we have Vinay give his presentation on his idea. This kind of ties into the GANs. We'll see how it compares and what we might be able to extend from this.
Yes, sir. I have a presentation. Can you?
Okay, can you put it up on the share your screen?
Yeah, everyone can see my screen, right?
Yeah.
Yeah. Okay. This is the paper that I found today. This is based on image synthesis, like creating new images based on, like we would show a set of images. Here in this case, it is the retinal images, and we would ask the model to generate more data of which looks like which looks like a realistic retinal images. So this was authored by P. Costa, A. Galran, and M. I. Mayer of the IN ESC Porto Institute. So let's get started on how they visualize this problem. So this is the contents that I have today. I'll be discussing about the aim and overview, and I'll go through the approach that they've discussed in the paper, and the model and implementation details, the images for results and experiments, and final the conclusion, like how what are the key takeaways from this paper that we can learn. So the aim, the overview. These are the retinal images that they've considered for this paper, like this one, and this is the corresponding vessel segmentation for this image. Like when, yeah.
Oh, I don't think anyone had any question.
Yeah. I heard. Okay. Sorry, these are the retinal images that they've considered for this paper, and this is the corresponding vessel segmentation. Like if you can see closely, these are the vessels that are highlighted in this. This is an image of our eye, and these are the veins these are the veins in our eye. So basically, they defined the problem as let us assume we have 5,000 of these images, these retinal images. We have 5,000, and if only 500 of them have labels like these, the rest 4,500 images are just unlabeled data, and they don't have a corresponding label like this. So how can we make use of this data set which has very less training labels, and the rest of the data set is completely unlabeled? So when we have these kinds of data sets, we would go with an approach something called as semi-supervised learning. So what this semi-supervised learning is about? It is nothing but making use of the training examples that we have, like the labels that we have, and we try to generate the labels for the unlabeled data as well, only with the help of the training labels we have, so that our entire data set will be having the labels, all the five. So basically, in this case, we'll try to make use of the 500 labels, image pair labels that we have, and we'll try to label the other 4,500 images that we have, 4,500 unlabeled images that we have. So that after that process, we'll have a 5,000 sets of image and label pairs. So that's basically how semi-supervised learning can come handy in this approach. So the aim is to generate realistic looking retinal images using a vessel segmenter and image-to-image translation of adversarial learning. So it's basically how we approach is basically in two steps that I'll discuss in the later slides. One is the vessel segmenter, and one is the GAN that we use, which is discussed in image-to-image translation paper. So
there's no change in the dots.
This is the approach.
Oh, yeah, go ahead.
I'm sorry, sir. No, thank you.
Oh, go ahead. Yeah, I was just...
Hello?
Yeah, you're okay.
Okay, sorry.
Yeah.
I was just breaking.
Oh, I don't think we had a question. Yeah, we didn't.
I think I'm having a network problem. So 1 second.
Okay.
Yeah. This is the approach that they discussed in the paper. So first, first, like we discussed, let's say we have 500 image double pairs, and rest of the 4,500 are just unlabeled data. So what we'll try to do is take a pre-trained model which can segment, which is a segmentation model, and we train a model to segment these images with the help of this 500 image double pairs that we have. So basically, after this step, we'll have a model which can segment those retinal images and extract those vessel paths out of those images. We'll do this with the help of a pre-trained model. We have some pre-trained models available. So there's something called a UNet that I'll be discussing in the later slides. With the help of that, we'll send our data, our 500 images of our 500 examples of image and label pairs, so that the pre-trained model will learn to extract those features, extract those vessel paths, and then we'll try to label that unlabeled data we have, that is the 4,500 images. So those are the 2 points that I've written here. And the third point is to make use of these 5,000 images of 5,000 images of retinal images, and then try to generate these kinds of images, which look realistic and are very close to what we have in the data sets. So let me discuss that in more detail. Obviously, we are using a conditional GAN over here. Like Bradley sir discussed, there's like a GAN can be conditional or unconditional. Nothing but a conditional GAN will be having labels. When we try to input labeled data into our GAN, that will be a conditional problem. And if we don't have any kinds of labels, and if we just want to generate images which look like our images in the data set, then that would be an unconditional GAN. As we have labels like these, and so we'll be using a conditional GAN, and that is the image to image translation. So this is the entire pipeline of the model. First, we have some labeled images. As we discussed, these are some, let's say these are some 500 pairs of real images and corresponding labels that we have. So we would take this would be a supervised vessel segmentation task. So we would take a pre-trained model known as UNet, and we would train the vessel segmenter, right? Now this vessel segmenter can take these unlabeled images, that are the 4,500 images, and can generate the vessel segmentations for these unlabeled images, right? So now we have 5,000 pairs of image and labels from this block. Now what we have to do is that with the help of these 5,000 image and label pairs, we'll try to generate more training samples with the help of GANs. So this is how it goes. We'll take these generated segmentation maps. We'll take the entire data set, actually, now the 5,000 labels that we have, and the real pairs that we have. Now here we'll use a generator and a discriminator. So the generator's task is to tell whether whatever the output that came from generator, does it look like a real or does it look like a fake image. So the task of a generator, what it will try to do is that it will take this it will take these vessel images, and it will try to generate the corresponding retinal images. So, and this discriminator will tell whether whether these generated images and these real images look alike or not. So the discriminator's task, the discriminator will then give a feedback to the generator saying that okay, there are some improvements that you can make, so that the whatever you generated will look more realistic. Then this generator will take the feedback from this discriminator and it will update the weights so that this generated images will look closer to these images, these real images, right? So basically how this happens is that first of all, uh, these these labels and these real images are shown to discriminator, and this will learn the difference between a fake image and a generated, like a fake image and a real image. Then this generator will will take each of this image, it will multiply this image with some weights that it has acquired during training, and then it will generate these images. So this discriminator will tell what to change so that these will look more realistic and more closer to these images. And likewise, this generator and discriminator will be iteratively trained so that they'll reach an equilibrium, they'll reach a state where the generator was able to bypass this discriminator test. So basically what I mean by that is that generator was able to generate an image which the discriminator couldn't identify it as a fake image. That means that generated image looks very realistic and very close to the original images. So that's how a GAN works. That's how this the relationship between generator and discriminator works. So so at the end of the training, the generator will be able to generate these realistic images with the help of these labels. Now now we can generate any number of labels that we can after the training. And then like going into detail of each of this block. First, let us discuss what really happens in this block. We will train a unit with the labels that we have initially, the 500 image label page that we have initially, and we would try to segment into an image looking like this. So as Bradley sir explained in his presentation, this is nothing but the encoder decoder model. This image is fed into patches into this encoder. Till this part it is called the encoder, and until this part it is called the decoder. So how this encoder decoder architecture will help us segment this image is that first the image will be taken as patches into this encoder. It will learn the representations, and it will extract a one single dimensional vector which has all the features embedded into it.
Yeah, there.
From here.
See there now?
Yeah, but nobody else is there. I was watching for cars.
Oh, okay.
Not gonna walk.
Yeah, and
Yeah.
and this decoder, what it will try to do is that it will take these extracted features, and it will try to reconstruct that image, reconstruct the image, but only highlighting the veins here. It should only highlight the veins here while reconstructing the image from this long vector. Now, how does this do is that it will take the ground truth image as a label. It will put this feature and this ground truth image as a side by side, and it will decide how to reconstruct an image from this feature vector so that it will look like this ground truth image. So basically, that is how the encoder decoder works, and that's how the training happens. And when we are done with when we send so many of these examples, the decoder will learn to reconstruct those images, reconstructed the images from these feature vectors. And then we'll be having a model which can take input images of retinal images and can output the vessel images. So that's the first part to first block we have, and this is how the GAN works. Let's discuss this in more detail now. So we'll take these label images, we'll send it into a generator, which is also an autoencoder, which is nothing but the encoder decoder model, and this is the encoder and this is the decoder. This encoder will extract the features from this image and we'll put it as a long feature vector here. So this vector will have all the features of this image. Now the decoder in this generator model, what it will try to do is that from this image, how can I reconstruct an image which looks something like this, right? That's the main job of generator. So it should take this image and it should output these kinds of image. So how does the generator know how to generate this kind of specific image? So that information will come from the discriminator. So first, initially, when we take fresh models, this image will go into the GAN. It will go into the generator, and it will produce some random noise, which doesn't look like a realistic image. And that realistic image, that generated image, will be sent to the discriminator, and this discriminator will decide whether whether the generated image looks like a real image or fake image, and it will provide feedback to the generator. Like it will provide a score on how realistic does it look like. And based on that score, this decoder, the weights of this decoder will get updated, and then the generator again tries to uh, generate images in the next iteration, and this training will keep on happening until um, so excuse me, can I go and plug my charger for a second? 1 second.
Yeah, go ahead.
Yeah.
Okay, Jesse. Yeah, thanks for attending. I guess Vinay's gonna finish up here, and I'll have the slides. I can make them available to everyone after the meeting. So thanks for attending. Hope you learned a lot. And okay, yeah. Okay, I think he's gonna come back in a minute here. So if you need to leave for after the end of the hour, fine. If not, you can stay. I think Vinay's going to finish up his presentation. Any ideas for the next blog post? I don't know yet. That's what I was thinking about. Probably an idea, maybe in GANs or something else. If someone has an idea, let me propose it. Send me an email or a Slack message, and we will talk about it. If I can get some interest in it, that's the idea. So yeah, but yeah, just you're welcome to suggest anything. Okay, yeah.
Okay, Dick had a comment in the chat. How about asking Thomas Harbeck to present his ideas on modeling Basilaria? Yeah, I think he should give a presentation on that. I can invite him. I talked to him the other day, and he's I don't know where he is. I think he's traveling, and he said he'll be available starting next week again, but or he'll be back on his regular schedule next week. So we should I should get him involved. I'll send him a message. Yeah, psychoanalysis of basilaria cells. Yeah, we should. Yeah, we should definitely look at that more closely. Actually, I had a conversation in the other group that I'm doing on like collective behavior, and we kind of went over that a little bit. And I think it's interesting that like people talk about like emergence and collective behavior, but they don't talk about like just other types of collective behavior that might exist. There could be things like cumulative behavior that isn't necessarily collective, or there's some sort of coordinated behavior. So I think that's worth talking about a little bit at least. Yeah. Oh, and Jesse, yeah, when I gave the talk on emergence, I don't know if you were there or not for that. I think you may have been there for that. It was the emergence talk where I talked about bees and swarms and birds and flocks. Oh, there's Vinay.
So
It's okay. We were just talking.
I ran out of my charging and I had to rush back to my room.
Okay. So you want to finish your presentation? We'll finish.
Yeah, it's the last 2 slides. Yeah.
Okay.
So I was explaining this GAN and the relation, how the generator model learns from the feedback of discriminator model and how it tries to fool the discriminator model so as to produce realistic images. That's basically what's in this diagram. And then this approach was taken from the image-to-image translation paper. It was published in 2018. Philip Isola et al. Published that. So I just took this image from this paper so as to.
Can you share your slides? I don't think they're being shared right now.
Oh, I'm sorry.
It's okay. There we go. Yeah, you were referencing something that wasn't on the screen, so.
I'm sorry, I forgot to share my screen again.
Oh, yeah.
Yeah. Yeah, previously I stopped at this slide, right? I was discussing about how the relation between this generator model and discriminator model. So this was wholly inspired by the image-to-image translation model, which was discussed, which was published in this paper by Philip Isola et al. 2018. I took this image to just provide one more example of how this GAN and how this GAN network works. Basically, what they're trying to do is that from this edge images, they're trying to replicate an object like this. So it's the discriminator's job to tell how good the generator is working, and it's the generator's job to fool this discriminator and convince the discriminator that whatever the generator generated, those images look like realistic. So in that paper, they actually made so many experiments based on image to label and label to image as well. One such example is this. So they trained a GAN on these pairs. This is a satellite map of a city of some buildings, and this is the label in this case. So from this, when this image is inputted into the generator after it has been trained, it was able to convert this image to this image. So that's why it is called image to image translation. Likewise, they've also experimented it the other way around. They put this image as input, like they swapped the data set, the elements in the data set, and then they trained the GAN on that, and it was able to translate from this image to this image. This is one example that they've shown, one experiment that they've done and published on the paper. Likewise, there are many examples as well in the paper. So I can put the link of the paper into the chat.
Okay.
So that you can refer to it later on. Also, it has many nice experiments listed. So this is how the results from the paper look like. So the top row is just the unlabeled data that we had initially. Like these are the images from the 4,500 images that we discussed earlier. And after training a segmentation model on the 500 images for which we have the labels, the model was able to generate the labels from this corresponding retinal unlabeled images. So this row corresponds to the labels of this unlabeled data, previously unlabeled data. Now, what we try to do is that we send these images into a generator, so that generator will be able to create an image which looks like a retinal image, realistic retinal image. So this row represents the final output that they've got. As we can see, we can infer that this is the real image. This is the corresponding vessel segmentation image, and this is the generated image from this vessel image. So it is possible that for a single label there can be two images as well, because if you can closely see in this generated image, the vessels are of the same pattern of this. So in that paper, they've concluded that for a single input we can have multiple outputs. Like for the same vessel we can have some other image which looks like a realistic image, but the inner vessel pattern is the same, like the inner label is the same. So that's what they concluded from that paper. So like I discussed in the earlier slide, this is the first point that tells the same. And then also another key point to take away from this paper is that these conditional GANs are great for semi-supervised problems. Like when I was searching for this paper, I've seen so much research, so many papers regarding this, how we can use semi-supervised, how we can use adversarial training in semi-supervised data sets or such, when we have such kinds of data sets. So this paper stands as a nice example for that. And that's it. That's what I have to present for this week.
Well, thank you. Yeah, that was good. I had a question from Dick in the chat. It's did they show row 1 minus row 3? And I think he's referring to those images that you had in the second to the last slide. Yeah, those.
No, sir. This is actually, this row 2 came from row 1. This row 3 came from row 2. So basically what we tried is that we generated, we took out the segmentation, we segmented this image, that is the result that is showing here, and from this segmentation we tried to reconstruct that image which looks like this image but not exactly this image. From this segmentation we tried to generate a image which looks like a retinal image but not necessarily the original image that from which this segmentation actually came from. So yeah.
Okay. Yeah, it was very good. Thank you. I think, yeah, we got a good idea of how this is implemented in a real data set. And they had some interesting approaches to, like, autoencoding, for example. I think that's good. So, yeah, again, this GANs area is very interesting. It's not very well, I think, applied to biological problems. It's not very well understood. So, and then, of course, Vinay wasn't here, but Jesse asked about another blog post, and I told him we maybe have some ideas from this set of presentations, maybe some other topics, but just keep in mind that we're looking for one more blog post idea. So thank you again, Vinay, for that.
Yeah, Vinay, my question is, I want to know how accurately row 1 and row 3 are equal, and you need to subtract them. If you subtract the images one from the other, you'll see the differences and whether there are any systematic ones.
Yeah, the discriminator tells us how relative, like how close this generated image and the original image is that. So the discriminator has a loss function to decide this. Basically, it would try, like as you said, it would maybe it could try subtracting this image from this image, and it would or it would extract the features from this image and extract the features from this image, and it would compare those features.
Yeah, I understand. I'm just saying you've got the original and a generated image. I just want to have a visual of how similar they are. And that's best done by making it, which is just the difference between them.
Sorry, sir, the voice broke. Can you please repeat it?
Oh, okay. I'd like to have an image that shows me the difference between each pair of images, because that will show whether or not there's systematic differences, which you should be able to see by eye. I mean, if they're
Yeah, I understood now. So
Okay.
basically trying to find out the difference between this and this, like how different they are.
right,
Right.
yeah, not by a number. I just want to see a picture.
Yeah, either by subtracting this image from this image or by any other means, like a relative relationship between these two images, right?
Well, the subtraction is a good way to do it, because then if there are regions, for instance, where there will obviously be some those errors uniformly distributed over the picture, or are they localized? You can answer questions like that simply by making a subtraction image.
Yeah, that makes sense. But in that paper, they did not put any metric for that, like how this and this will compare. These two will compare. But I think we can do such thing. Like we can take this generated image and we can take this image and we can try to analyze the features of both of the images and see how, like we can put some matching algorithms or comparison algorithms that we have over features.
Yeah, okay. Well, I'm just saying visual comparison is sometimes the best, because if you simply do some measure of how similar they are, the measure doesn't tell you much about the difference image. Can you get a hold of the original 512 by 512 images?
Oh yeah, in that paper they have given the link to the dataset. I haven't downloaded the dataset yet. So
yeah, if it wouldn't take you too long, it'd be nice to see the differences between them and see if see how they look.
yeah, sure. I think they actually gave the link to reproduce these results as well, because I think they gave. Let me try to find the link again. They gave this link to reproduce the results, so that also they'll give a link to where to find this data set and all. So maybe we can try and see, we can replicate the results ourselves, and then we'll have this image and this image in our system. So then we can try to see.
That's a lot more work, but okay.
Yes, I'll try to find out the link for that, and I'll post that in this in the group today tonight.
Okay.
Yeah. So, and yeah, any other questions?
I don't have any other. Jesse, do you have any other questions about the paper? I'll get all the links together and send them out. So
let me give you an example where even in this case it would be very important. Sometimes people can develop tumors in the eye. Okay? And suppose that you wanted to detect a tumor at a very early stage, then you would like to see different images to see if a tumor. Okay. Here, I can give you an example which I published on this stuff. Okay, take a moment here for me to dig it out of the computer.
okay.
Okay. Chat. Oops. Okay, here's the reference.
Okay.
Can I transmit a file?
No, we have to.
Oh, okay.
Yeah.
I'll send you the files. It's in my papers. This would be under the medical imaging section.
Okay.
Okay, so you probably already have it in your computer. Do you know what Waldo is?
Oh, Waldo. Where's Waldo? Yeah.
Other guys know it. I don't know where's Waldo got to India or not. Did we lose Varma?
Oh, I think. Yeah, I don't know. He's been having trouble with his computer. We might have lost him, but. Well, I can post the paper.
Okay. At any rate,
Yeah.
this is a paper on how to find Waldo.
Okay, yeah. In a mammogram, not a actual Waldo.
Well, no, I actually use Waldo.
Okay. Well, I mean, yeah.
Waldo is in the paper. You know,
Okay. Yeah.
I'm just saying that these techniques, you see, the techniques are generating, comparing images, but if you have a time sequence of images, then you can start to detect things like the beginning of a tumor or a beginning of blood vessel problem, stuff like this.
Okay.
Okay? So these techniques can be extended to the time domain.
Yeah, that sounds pretty good. Well, thanks again for presenting, Vinay, and we're probably at the hour and a half mark, so
Yep.
I have to cut it